Hello and welcome to chapter 28, chapter 18. We are going to go over this chapter with two lectures. So I'm just going to go over the first half of chapter 18 in this lecture, and then I'll post another one on Thursday with the remainder of this chapter. So this chapter is about the cell division cycle, so cell proliferation. So there's three main questions you can ask about cell proliferation. First, how do cells duplicate their contents? Second, how do they partition these duplicated materials and split in two? And third, how do cells coordinate all of these steps, all the proteins and enzymes that are needed for both of these processes? So we've got duplicating the DNA, we've got splitting the DNA in two, but then also the rest of the cell has to get split in two as well. And so in chapter 18, we are going to focus on these second two questions because number one, it's kind of been done to death, right? You know the cell cycle, you know the stages of mitosis, right? How DNA replication works. So we're going to focus more in detail on how exactly the cell divides up the DNA and also how the cell cycle coordination works. So the objectives for this chapter are first, to refresh your memory on the stages of the cell cycle. Second, to know how it is controlled. So what are the different types of regulator proteins that are involved? What are they checking at each stage in the cell cycle? And third, we're going to talk about specifically M phase, the chromosome duplication, so that occurs during S phase, and segregation of DNA, which occurs during M phase. Lastly, we're going to talk about extracellular signals. So what signals tell a cell to divide in the first place? And how do you get from a signal outside of the cell to DNA being copied and the cell actually dividing? So first, let's go over the cell cycle. We have during the majority of the cell's life, right, or a good portion of it is just the cell growing in size, acquiring its mature phenotypes, and accomplishing its functions that it's responsible for. And at some point, the cell during this phase of life is going to get a signal that it needs to divide. So the first thing that it's going to do before the cell divides is it's going to duplicate its DNA. Because if the cell does not do this, it will split its DNA in half, and then there'll be only a quarter remaining, right, and so forth. The DNA would eventually just be split down into nothing. And kind of like when no one wants to eat the last piece of the donut, right? They split in half and then someone else takes a quarter, right? The infinity donut. So the cells have to first replicate their DNA, and that occurs during S phase. Next, the DNA has to get separated. Those two copies need to be separated or segregated to opposite sides of the cell. And then the remaining part of the cell, the cytoplasm and everything in there, has to get split into these two daughter cells. So proliferation, that's how cells reproduce, how they make copies of themselves, and it's just the cycle of DNA duplication and cell division. And you may not have thought about this before, but we only talk about DNA normally. Well, other things get duplicated in the cell as well. We've got all these organelles, right? So this is the fun part about cell biology, where you get to learn more of the nitty-gritty of what's going on in the cell. Another thing to think about is that division is coordinated with growth. What I mean by that is that every time a cell divides in two, each resulting cell is half the size of the original. And so the cell needs time to grow back up again in size before it can divide again. Otherwise, each cell will just get smaller and smaller and smaller and smaller. And so the cell has to check and see, have I grown large enough? Am I ready? Do I have everything made that I need to in order to actually divide again? And so there's cell cycle checkpoints that help to coordinate growth and cell division. And while cell cycle, length of cell cycle and the details of it exactly, what specific proteins are involved may change across species, but in general, there's many features that are quite common and that's what we're going to focus on today. So just an example of some variety here, looking at cell cycle times. So during development, cells divide quite rapidly. Early frog embryo cells divide every 30 minutes. By zebrafish embryos, the cells divide about every 15 to 20 minutes. So you can check on the cells over the course of the day and watch it start from a single cell, ending with like 1,000 cells, right? Within just a day, which is pretty cool. Or no, more than 1,000 cells. That's right. But anyways, it's fast. And yeast cells also pretty fast. Now, when we look at things like fibroblasts, those are going to be a bit slower. They're still proliferative, but it's about once a day. You can see that this other adult cell type, intestinal epithelial cells, these divide about every 12 hours, so about twice a day. So that's rather rapid for an adult cell. And because it divides so frequently, the intestinal epithelium is being replaced constantly. And so when you have treatments like cancer drugs that affect fast-growing cells, you know about hair falling out, right? Because hair is fast-growing. Well, so are these intestinal epithelial cells, and so the loss of those cells is what results in those side effects of nausea. Now, as a reminder, eukaryotic cell cycles consisting of 4 phases. We have G1, S, G2, and M phase. So M phase involves mitosis, which is the literal division of the DNA, and cytokinesis, which is division of the cytoplasm. Cytoplasm. Kinesis, movement. And the rest of the cell cycle collectively can be called interphase. So anything that's not M phase, whenever the cell's not actively dividing, it's said to be in interphase. And this is when things like cell growth occur, where the cell accomplishes its major functions, and also when it copies its DNA in preparation for dividing. Now the cell, again, like I said, during this interphase, during G1 specifically, it's growing in size, and the cell has to make sure that it's reached a ready point before it can go into DNA replication or M phase. So let's stop and do a problem. This problem involves a technique called fluorescence activated cell sorting, or FACS, or flow cytometer is another. It's basically a machine in which cells are shot through in front of a camera, one cell at a time, and their fluorescence is measured, right? So in this example problem, we have cells that are stained with a dye that becomes fluorescent when it binds DNA. Okay, so more DNA, the more fluorescence there is. And what the cell does is it measures the intensity of the fluorescence in every single cell and then graphs it. Here you have on the y-axis, you have the number of cells, and then on the x-axis you have relative intensity of fluorescence. So you can see that we've got one group of cells where a large proportion of the cells have a low level of fluorescence. And then we have another large portion of cells in B that have very high levels of fluorescence. And then we also have a group of cells that have kind of a moderate level of DNA or fluorescence. Right? So these cells are proliferative, so they could be at any stage in the cell cycle. And I'd like you to take a minute to indicate where on this graph is representing cells that are in G1, S, G2 phase, or mitosis. And then second, to ask yourself, which phase do you think is the longest in these cells? So as you're doing this problem, remember that the amount of fluorescence is directly proportional to the amount of DNA in the cell. So think about, when would a cell ever have more DNA or less throughout the cell cycle? So pause this video, take a minute to try and do this problem. Understanding how to do this problem and read this figure is quite important for this chapter, so I encourage you to study this problem in preparation for our last exam. So let's take a look at this figure. So we've got some cells that have a lot of DNA, some that have less, and some that are kind of in between. Now, the amount of DNA in a cell is going to double during which phase? It should double the DNA amount should double during S phase, because that's when the DNA gets duplicated, right? When in the cell cycle would the DNA drop back down again? So if you think about it, during S phase, the DNA gets duplicated. During G2, that cell has twice as much DNA. During M phase, it gets split into two. And at the very end of M phase, we have two daughter cells, and now they're back at that original amount of DNA. And you can see that in peak B, it's approximately double the intensity of fluorescence as in peak A. So we can first say that we know pretty well that peak B is going to be any time after the DNA gets duplicated, but before the cell divides. Right? And so at what point would the cell have only one copy of DNA? That would be during G1 phase. Right? The cell has not duplicated its DNA yet. When might it have moderate in-between amounts of DNA? I would say probably during S phase, as the cell is starting to begin DNA duplication, but it's not quite finished. Right? So again, most of the cells, as shown in peak A, have a lower amount of DNA. And then we've got a smaller portion of cells that have about twice as much as DNA in peak B. And the smallest fraction of cells have a moderate amount of DNA. So we said that peak A was G1 phase. This is cells that only have one copy of DNA. Right? The cell has not duplicated its DNA yet. And that during S phase, the cells copy the DNA and duplicate it, so that by the time you get to G2, you have twice as much DNA. And so that's what we're seeing in peak B, is G2 as well as M phase, right? Because it's not until the end of M phase that the DNA actually is split into those two daughter cells. So that range in between is going to be cells that are in S phase because they're starting to copy their DNA, so there's more than normal, but they're not done yet, so there's not twice as much DNA. Now, to determine which phase is the longest, that's just going to be indicated by the number of cells. The longer a phase is, the more likely you're going to capture cells in that phase, right? So if you guys took Principles of Zoology here at Weber State way back in the second lab of the semester, if you got to do cell counting under the microscope, we did a problem experiment kind of like this, right? The longer the cell cycle stage, the more likely you are to capture the cell doing that, right? And so that is why we're going to say that in these cells that G1 is the longest phase. But a way you could study using this problem is you could say, all right, what would this graph look like if G2 was the longest phase, right? Or what if G1 phase was relatively short and about the same amount of time as the rest of the phases? What would that graph look like? What would that imply about the cell's behavior and their proliferation rates? Let's move on to cell cycle control. So we've got a network of regulatory elements that are all kind of interacting, and the main job of this system is to make sure that the phases occur in the correct order and that you don't move on to the next phase until the first phase has been done completely and properly. So you don't want to be jumping back and forth from G1 to S and then back to G1 again and then to M, and you don't want to divide the cell until the DNA gets duplicated in S phase, right? And so what the cell does is it has this feedback mechanism, and so it checks. For example, before M phase, before the DNA gets divided into two daughter cells, it has to be duplicated, right? So there's a system in place in which proteins sense, has the genome been duplicated or not? And if not, the cell will pause in the cell cycle and will not continue until that occurs. So these are checkpoints, and there are There are three main points in which the cell cycle is regulated, in which the cell has this feedback mechanism. It checks in and says, is everything going well? If not, let's pause. If yes, let's keep going. And so you should know what these three checkpoints are, what things they're assessing to determine whether or not the cell can proceed through the cell cycle. So first, when you have a cell hanging out in G1 phase, it's going to receive a signal from the environment that tells it to divide. And so at this checkpoint, the cell cannot proceed into S phase unless the environment is favorable and things like the cell's grown large enough in size, as we talked about before, and it has all of the correct proteins and machinery synthesized to begin on to the next stage of the cell cycle. So the cell copies its DNA during S phase. In G2, it prepares to enter mitosis, enter M phase. Now, the cell has a second checkpoint here. It's saying, did all the DNA get replicated, right? If all the DNA hasn't been duplicated, we do not want to start dividing the cell in half. And second, if any DNA has been damaged, we want to make sure that that gets fixed because we do not want to be passing down damaged DNA to daughter cells. That's a good way to get cancer. So only once those questions have been answered positively will the cell then leave G2 and go into M phase. And partways through M phase, we have our third checkpoint, and this occurs during metaphase. So during metaphase in mitosis, the cells, the DNA has been all lined up in the center of the cell, which we'll talk about in some future slides, right? And so they are going to be pulled apart. The copies are going to be pulled apart to opposite sides of the cell by the cytoskeleton. And so we need to make sure that that DNA in the center of the cell is attached correctly to the cytoskeleton so that everything's being evenly split in half. We don't want something to accidentally, say you have both sister chromatids go to one side of the cell and not be evenly split in two. And so that is our last point in which the cell checks and says, all right, if all the chromosomes have been properly attached to the mitotic spindle and aligned in the center, we're good to go to literally pull that DNA apart opposite sides of the cell and proceed through the rest of M phase. All right, because that's kind of a point of no return, right? So it's important to have a checkpoint there. Now, the proteins actually involved in this feedback and this sensing mechanism and telling the cell that it's ready to go on to the next stage of the cell cycle are called CDKs, or cyclin-dependent kinases. And so as you can guess by their name, they phosphorylate things, and their activity is dependent upon another protein called cyclin. And cyclin levels will rise and fall throughout the cell cycle, hence they will cycle, and that's why they're called cyclins. In this cartoon, you can see a cyclin-dependent kinase. When it gets activated, it tells the cell it's ready to go on to the next stage of the cell cycle. However, it won't be activated unless it's bound to this protein cyclin. So we have some different types of cyclins and the cyclin-dependent kinase that together form these complexes. And so you should know these complexes. So first we have M-cyclin, right? This binds to M-CDK. Next we have S-cyclin, binds to S-CDK. We have G1S-cyclin, binds to G1S-CDK, and then G1-cyclin that binds to the corresponding CDK. Okay, so keeping track of all these, M-cyclin and M-CDK, those are important for telling the cell it's okay to move to M phase, right? Or move through M phase. The S-cyclin and the G1S-cyclin, those are the ones that tell the cell it's okay to move on into the S phase. So again, when I talk about cyclin levels rising and falling, this is a nice diagram from your book that shows this. So we have some cells here that are going through division 3 times, and we have M-cyclin levels rising and falling, rising and falling. So M-cyclin will bind to MCDK, induce the cell to undergo mitosis. So only when M-cyclin levels are high enough will there be enough cyclin to bind to its corresponding CDK, activate that CDK. So it says that MCDK activity is rising and falling. But the actual protein levels of CDK are constant. All right, so just because a protein is high in concentration doesn't mean that it's active, right? So this MCDK, or this MCDK is present consistently throughout the whole life of the cell, but it's not necessarily active until cyclin binds to it. And cyclin has to get high enough concentration in order to have enough cyclin to bind all the CDKs. Once that happens, these CDKs are active, and it tells the cell it can proceed through mitosis. And so you can see, right, only when M-cyclin levels are high does the cell proceed through mitosis. So again, we've got protein levels rising and falling, right? These cyclins are cycling. And in this example, we can see that M-cyclin starts to rise during G2, and partways through M phase it drops, and that S-cyclin rises partways through G1, actually stays up for quite a long time, doesn't drop till M. And it's really important to be able to regulate the levels of these proteins. Right? So first of all, they can be regulated by how much is made, right, at the level of transcription. But we've also talked about other ways of controlling protein levels, and that other way is by breaking down proteins, so by reducing their levels, by increasing the amount that are getting digested, that are getting graded. So how exactly would you reduce a cyclin? Right? If you want to do this very quickly. So to break down these proteins, to degrade these proteins, we have a complex called anaphase promoting complex, or APC. This is an important one to know. We're actually going to bump into it again later in the lecture. So what APC does is it directs the cyclin towards proteasomal digestion. Right? So we know that proteasomes break down proteins and other molecules in the cell. So in order to turn off this signal, the cyclin levels need to drop. And so to get rid of cyclin, anaphase promoting complex will put these ubiquitin tags on cyclin, and that targets it, tells the proteasome that it needs to be broken down. In addition to cyclin having to bind CDK, phosphorylation is a second level of regulation for CDKs. So again, cyclin rises gradually. However, the activity of this cyclin CDK complex, it does not turn on gradually, right? It's on or it's off. And so we talked about phosphorylation as a molecular switch. It's a great way to turn on or off very decisively. So in this case, phosphorylation inactivates these cyclin CDK complexes. We've got CDK present but not active consistently throughout the cell cycle. And then we've got cyclins rise. There gets to be a point where majority of the CDKs are bound to cyclin, but they're not yet active until they get dephosphorylated. And then, boom, you've got a lot of CDKs that are all active at once. And the way this works is if you look on the right side of this picture, we have a phosphatase. Right? Phosphatases are what dephosphorylate things. Kinases are what add phosphate groups. So and when the cell is ready, the CDC25 phosphatase will remove that inhibitory phosphate to allow that MCDK to be active. And when it needs to get shut off, there's a phosphorylation event by this kinase, pretty fun name, WEE1. In addition to phosphorylation, we also have inhibitor proteins. So in this example, we have P27. This protein is giving like a big hug to this cyclin CDK complex and not letting it do anything. Right? So in addition to phosphorylation inhibiting cyclin CDK complex, you can also have binding of inhibitory proteins such as P27. So where do these all fit into the cell cycle control that we've been talking about? At the beginning before the cell can go into S phase, we have CDK inhibitors. So throughout all of G1, these cyclin CDK complexes, if there are any, they are being kept turned off by inhibitory proteins. They're just binding and hugging, right, to those. And so when the cell is ready to go into S phase, these P27 or these CDK inhibitors get removed. And so now we have these active cyclin CDK complexes that tell the cell is ready to move on through S phase. Now the DNA has been duplicated, the cell's proceeding through D2, and it wants to enter into M phase. And so at this point, phosphorylation is an important regulator of these cyclin CDK complexes. So we have the cyclin CDK complex that is responsible for allowing the cell to enter M phase, and it is being inhibited by phosphorylation. All right, and basically not until this activating phosphatase gets turned on will that CDK complex be dephosphorylated, activating it and telling the cell that it can proceed into M phase. And then at M phase, we talked about how during mitosis, at metaphase, the DNA lines of the chromosomes line up at the center of the cell. And only when those cells are lined up properly and the DNA is connected to the mitotic spindle properly and evenly will the cell proceed into anaphase, the next stage of the cell cycle. And so the regulator there is that anaphase promoting complex, right, APC. And so what APC does, if you look back at that slide again, it is going to promote the cell to continue through mitosis, go into anaphase. And so normally that promoter is shut off, but when the cell receives all the correct signals that things are good to go, that APC will be activated, and it will then allow those CDKs to be activated to allow the cell to proceed through the rest of M phase. So take some time, look at these different regulators, go back and forth through these slides to see which ones are regulating which stages of the cell cycle. Let's talk about the third objective, focusing on chromosome duplication and segregation. So as we said previously, during G1 phase, the cell is growing. It may be performing its cellular functions, and it's getting ready to receive a signal that it needs to divide. So at this point, the SCDKs and MCDKs are not around, right? They're very low levels. Either the cyclins are not being made, or these CDKs are bound by inhibitors, right? So cells can hang out in G1 for quite a long period of time. You may have heard of G0. This is a resting state in which cells are saying, you know what, we're never going to divide again, so we're just going to pack up all of our cell cycle machinery and put it under the bed. Right? Now, some cells, though, they may be ready to go, and so when they receive a signal, the cell gets ready to move on into S phase. And so these signals that induce cell division are called mitogens. And we talked about mitogens in the last chapter. These are going to induce signaling pathways that ultimately result in changes in the levels of cyclins, right? So this is going to induce cyclins that tell the cell to proceed on into S phase. Let's talk about an important protein called RB, or retinoblastoma. This is an important checkpoint protein, and it is going, the way it functions is it is going to bind to transcription factors inhibiting expression of particular genes important for cell proliferation. So, retinoblastoma, this protein was discovered in cases of the certain retinal cancers. That's why it's called retinoblastoma, but its real function has to do with a variety of cell types, but not just retinal cells. Its main thing it's important for is regulating cell cycle. So what happens is that normally these genes that are responsible for telling the cell to divide are kept silent by this RB, right? We know that transcription factors need to bind to promoters to tell a gene to be expressed. And so what RB does is it binds these transcription factors and keeps them from assembling at the promoter. So when the cell does receive a signal from a mitogen, there's going to be the cell signaling cascade, and ultimately these G1 CDKs and G1S CDKs are going to become activated, and what they're going to do is phosphorylate this RB protein. Phosphorylation forces RB to let go of these transcription factors, so now they can turn on expression of genes that are important for cell cycle proliferation. Here we have a cartoon on the right. You can see a mitogen binding to a cell receptor, and that once there's a downstream signaling cascade that ultimately is going to end up with activated cyclin-dependent kinases. And what these are going to do, kinases, they're going to phosphorylate. And their target is RB. So normally, when the cell's not receiving a signal and it's just hanging out in G1, RB is bound to transcription factors, keeping them inactive and preventing the expression of genes that help the cell progress through the cell cycle. When this mitogen signal is received, that causes RB to become phosphorylated, and in this case, phosphorylation causes a slight adjustment in the conformation of this protein, making it let go of the transcription factors. And then these transcription factors combine and activate expression of cell proliferation related genes. Another thing that the cell needs to check for is that the DNA that's being replicated and being passed down to daughter cells is not being damaged. And so the cell has this feedback mechanism, or a sensor, where DNA damage causes induction of this protein called P53. And P53 is responsible for turning on or turning up levels of protein P21. P21 is a CDK inhibitor, right? So it's one of those ones that binds to a cyclin CDK complex, prevents it from performing its functions. And so the more of the P21 that you have, the less likely the cell is going to proceed through the cell cycle. So if you have DNA damage, ultimately that's going to lead to more inhibition of these checkpoint proteins. It tells the cell, stop, hold up, wait a minute. We do not need to go on to S phase quite yet, right? So this is specifically inhibiting G1S CDK and SCDK, preventing entry into S phase. So in this diagram, you can see, summarizing what I said, say you have X-ray or sun damage, whatever it is, DNA gets damaged, it is detected by P53. So active P53 is going to act as a transcription factor to turn on P21 expression. So P21 protein is going to get made, and this protein is going to inhibit certain CDK complexes, specifically the G1S and the SCDK complexes. And so they cannot tell the cell to go on into S phase because they're being inhibited by this P21. This gives the cell time to repair its DNA before it gets copied, And if the damage is bad enough, it can actually induce cell death by apoptosis. RB is super important. It has mutations in RB are seen in cancers, as well as mutations in P53. Right? So very important proteins, why we're talking about them today. And you can see why regulation of the cell cycle is so important, right? Because if it's not, you get cancer. And so there's layer upon layer of regulatory mechanisms and checkpoints and feedback to control progression through the cell cycle. So again, I talked about G0 phase, right? This is when the cell is in this resting phase where it's not going to divide for a while. However, it can ultimately shut everything down and never divide again. So an example of that would be the majority of the neurons in your brain are not proliferative, and so they are going to undergo terminal differentiation. They're never going to divide again. So let's look at an example in real life of a mitogen inducing cell proliferation. Okay. So I used to work with neural progenitors, so brain stem cells in dishes in my previous lab. And what we had was these cells, they're neural progenitors. They can become neurons, astrocytes, or oligodendrocytes. And if you want to study them as stem cells, you want to keep them proliferative. You don't want them to start maturing into the different final states. However, if you want to induce this differentiation, if you want them to mature, we would change what was in the growth media, right? And so mitogen withdrawal, the statement, it says mitogen withdrawal induces terminal differentiation. So just to take a minute to talk about that. So first of all, the cell can be dividing or it can be maturing, right? It can't be doing both. You know, try to imagine a neuron sending an action potential while it's in the middle of cytokinesis, right? It's not going to happen. So cells have to be either, especially stem cells, they're going to be either dividing. If they're not dividing, that gives them time to differentiate and to mature into, in this example, neurons, astrocytes, or oligodendrocytes. So when we want to maintain our cells as stem cells being proliferative, in their cell culture media, we include growth factors or mitogens. And when we want them to stop proliferating and to mature into neurons, astrocytes, or oligodendrocytes, when we want them to undergo terminal differentiation, we will remove that mitogen, take it out of the media. So if you look on the left, this is an example from a publication where they looked at levels of cell proliferation as indicated by BRDU labeling. And you don't need to know how BRDU labeling works, just that the more BRDU labeled cells there are, the more cells are proliferating. And so when they have the cells in the regular conditions control, you can see that about 20% of the cells are dividing or BRDU positive. And when they remove the growth factor, when they remove the mitogens, in this case, it is fibroblast growth factor or epidermal growth factor. When they remove that, the levels of proliferation start to drop, right? So if the cells are not dividing as much, and these are stem cells, that means that they're going to start maturing, undergoing terminal differentiation. And so that's what we can see on the right. On the x-axis in figure B, you can see days after FGF withdrawal. So days after they've removed that mitogen, we can see markers of mature cells, neurons, astrocytes, or oligodendrocytes rising, such as MAP2A, which is a marker of neurons, or GFAP, which is a marker of astrocytes, and markers of proliferative stem cells, pluripotent stem cells, such as nestin and prominin drop. Right? Pretty neat. So this is just a side note I wanted to take to talk about really the importance of mitogens and to get you a little taste in stem cell research, which is pretty fun. All right, so we've got G1 phase. The cell has got a signal from a mitogen, and it is ready to go on to S phase. All right, so again, before a DNA can divide, the DNA must be duplicated. It must be done correctly, right? And so SCDK is what's going to promote entry into S phase and that DNA copying. And it also prevents re-replication. So we don't want the cell to start making quadruple levels of DNA, right? It just needs to duplicate it once. So early during G1, there will be proteins that will start to get assembled to prepare for DNA duplication. And one of these proteins is called origin of replication, or ORC. And I always get a little Lord of the Rings reference here. And what happens with this ORC is that it recruits a protein called CDC6. And ORC, along with CDC6, are going to load a helicase onto the DNA. Now you should know what a helicase does, right? Helicases are important for separating duplexed DNA, and that's important for things like DNA replication and transcription, right? And so we're really getting into the nitty-gritty, right? How does DNA get duplicated? So this is all actually starting right during G1. The cell's getting prepared for the DNA is getting prepared to be copied. And then we also have formation of this pre-replicative complex that is a variety of proteins and enzymes that are assembling at the DNA. Then at the end of G1, SCDK gets activated and the cell can proceed into S phase. So this helicase has just been sitting and ready to go until SCDK becomes activated. SCDK allows that helicase to work to unzip the DNA, and the remaining proteins that are needed for DNA to replicate are assembled. So we've got a graph or a picture of this. We've got during G1 phase, we have our origin recognition complex, or ORC, sitting on the origin, which is basically the site where DNA duplication begins, and it's in complex with CDC6. Those CDC6 and ORC help to recruit the DNA helicase, shown in yellow. And then when SCDK levels rise sufficiently and when S-cyclin and CDK complex is activated, that helicase is told that it can start unzipping the DNA. Right? In addition, the rest of the replication machinery, such as DNA polymerase, are recruited. The polymerase is what actually copies the DNA, right? Creates our duplicate strand. Now, if replication does not occur correctly, the cell can get arrested in G2, or basically frozen in G2. Again, this is to make sure that everything is done correctly and any mistakes are fixed before the cell divides. And we talked about this before. So entrance into the M phase is regulated by M-CDK, M-cyclin CDK complex. And this is one of those situations where the enzyme is regulated by phosphorylation. So the cell will not proceed on to M phase until M-CDK gets dephosphorylated by this activating phosphatase CDC25. And so when DNA is damaged or not correctly replicated, CDC25 will not function, right? So only when the cell has correctly replicated its DNA will CDC25 be activated. And this, what else do I want to say? Once it's activated, then the M-CDK gets dephosphorylated, and the cell is told by this M-cyclin CDK complex that it can move on into M phase. But until that happens, until the DNA is fixed, that M-CDK is going to remain inactive. So again, during M phase, this is when we have mitosis and cytokinesis, and also reorganization of the rest of the cell. So a lot of the material in the cell, organelles and things like that, are going to be distributed evenly among the daughter cells. And then, of course, we've got the mitotic spindle that helps us separate the chromosomes. So MCDK, again, we talked about how that's important for cells to exit G2 and go on into M phase. And it gets activated by dephosphorylation. We talked about that before, right? Interestingly, once MCDK gets activated, it can actually perform positive feedback, kind of some self-regulation, in which it can continue to activate its activator. Right? So MCDK is a cyclin, or is a kinase, right? Cyclin-dependent kinase. So in addition to phosphorylating its target proteins downstream to tell the cell to go through M phase, it will also phosphorylate its activator, CDC25, which is a phosphatase. This phosphatase, remember, it is inactive until it becomes phosphorylated. Also, it can shut down this inhibitory kinase Wee1. So Wee1 is what inhibits or turns off MCDK. So it can kind of prevent its own inhibition as well. Now, one thing that I think takes a while to really comprehend is really how important DNA packaging is in various stages of the cell cycle. For example, we've talked about heterochromatin and euchromatin. We've talked about how DNA gets packaged in the cell, right? And a lot of times when we think of pictures of DNA, right, we see them as these two chromosomes kind of attached to each other. That is only seen during mitosis, right? The cell, or during M phase. The majority of the time, the DNA is all loosey-goosey, kind of mixed up and intertwined amongst itself, spread out throughout the inside of the nucleus. And so only when you need to organize DNA does the DNA start to condense and become more compacted. I like to use the example of if you've got a basket of yarn of different colors, right? If you let your cat get in there, it's going to get all mixed up and tangled. And so if you want to use just the red yarn, you're going to have to wind it all up into a nice tight bundle. And that allows you to easily grab whatever color you want out of your basket. Same thing with the DNA. It only needs to be condensed like this into tight packages during M phase when we are separating these chromatids to opposite sides of the cell and organizing the DNA along the center of the cell, things like that. And so to get a little deeper into how this works, we're going to talk about cohesins and condensins. So again, these help to, as you can guess by the name, condense the chromosomes during M phase. So here we have a picture that indicates how condensins could work. So yellow is our DNA, and it's already in those yellow strands, it's actually already been packaged into these nucleosomes that are all wrapped around each other as well, right? So we're kind of looking at a larger scale. These condensin proteins are going to help to kind of package the DNA up in a more organized and tight manner, again, to allow easy separation. And we talked about how MCDK, it's a kinase, and how it targets different proteins to tell the cell to proceed on into M phase. So condensation of DNA, making the DNA more compact, is regulated by MCDK because this condensin protein will not function unless it gets phosphorylated. So it's pretty neat to be able to kind of connect the dots of what's happening in the cell. Again, this picture here, just to demonstrate, right, this is normally only seen during M phase, having the DNA condensed like that. And you can just disregard the arrow, it's not important. So the DNA gets duplicated, right, during S phase, and each copy is called a sister chromatid, as a reminder. They remain tightly bound together, right? You see those two chromosomes very close to each other in that previous picture. So we talked about condensins. We have another protein called cohesin. Cohesin being cohesive, right? It holds those two sister chromatids together. And in late mitosis, those chromatids are pulled apart, right? And the way that works is by breakdown of those cohesin proteins. So now that the DNA has been condensed, we've got the mitotic spindle forming out of microtubules. It will separate those duplicated chromosomes so that one sister chromatid is on each side of the cell. In the next lecture, we'll go through the stages of mitosis and talk more about extracellular signals that regulate cell growth and division.